In either case, the migrant beetle will reproduce with the local population. This will result in the genes of the migrant beetle entering a new population. This kind of gene flow is bound to happen between populations that are part partly but not completely separated. If, however, between two such subpopulations a large river comes into existence, the two populations will be further isolated. The levels of gene flow between them will decrease even further. Over generations, genetic drift will accumulate different changes in each subpopulation. Also, natural selection may also operate differently in these different geographic locations. Thus, for example, in the territory of one subpopulation, crows are eliminated by eagles, but this does not happen for the other subpopulation where crows numbers are very high. As a result, the green variation will not be selected at the first sight, while it will be strongly selected at second. Together, the processes of genetic drift and natural selection will result in these two isolated subpopulations of beetles becoming more and more different from each other. Eventually, members of these two groups will be incapable of reproducing with each other even if they happen to meet. There can be number of ways by which this can happen. If the DNA changes are severe enough, such as a change in number of chromosomes, eventually the germ cell of two groups cannot fuse with each other, or a new variation emerges in which green females will not meet meet with red males but only with green males. This allows very strong natural selection for greenness. Now, if such a green female beetle meets a red male from the other group, her behavior will ensure that there is no reproduction between them effectively. New species of beetles are being generated. Evolution and classification. Based on these principles, we can work out the evolutionary relationships of the species we see around us. It is a sort of going backwards in time. We can do this by identifying hierarchies of characteristics between species. In order to understand this process, let us think back to our decision in the classification of organisms in class 9. Similarities among organisms will allow us to group them and then study the group. For this, which characteristics decide more fundamental differences among organisms and which one decide less basic differences. What is meant by characteristics? Anyways, characteristics are details of appearance or behavior. In other words, a particular form or a particular function. That we have four limbs in the thus of character. That plant can photosynthesis is also a characteristic. Some basic characteristics will be shared by most organisms. The cell is the basic unit of life in all organisms. The characteristics in next level of classification would be shared by most but not all organisms. A basic characteristics of cell design that differ among different organisms is whether the cell has a nucleus. Bacteria cells do not, while the cell hmm, while the cells of most other organisms do. Among organisms with nucleated cells, which ones are unicellular and which ones multicellular? The property makes a very basic difference in body design, but because of specialization of cell types and tissues among multicellular organisms, whether they can undertake photosynthesis or not will provide the next level of classification. Among the multicellular organisms that cannot do photosynthesis, whether the skeleton is inside the body or around the body, will make another fundamental design difference. We can see that even in these few questions that we have asked, a hierarchy is developing that allow us to make classification groups. The more characteristics two species will have in common, the more closely they are related. And the more closely they are related, the more recently they will have a common ancestor. An example will help. A brother and a sister are closely related. They have common ancestors in the first generation before them, namely their parents. A girl and her first cousin are also related, but less than the girl and her brother. This is because cousins have common ancestors, their grandparents, in the second generation before them, not in the first one. We can now appreciate the classification of species is in fact a reflection of the evolutionary relationship. We can thus build up small groups of species with recent common ancestors, then supergroups of these groups with more distant common ancestors, and so on. In theory, we can keep going backwards like this until we come to the nation of single species at the very beginning of evolutionary time. If that is the case, then at some point in the history of Earth, non-living material must have given rise to life. There are many theories about how this might have happened. It would be interesting to come up with this theory of your own. Tracing evolutionary relationships. When we try to follow evolutionary relationships, how do we identify class characteristics as common? These characteristics in different organisms would be similar because they are inherited from a common ancestors. As an example, consider the fact that mammals have 4 limbs, as do birds, reptiles, and amphibians. The basic structure of limbs is very similar, though it has been modified to perform different functions in various vertebrates. Such a homologous characteristics helps to identify an evolutionary relationship between apparently different species. However, all similarities supply in organ shape are not necessarily because of common ancestry. What would we think about the wings of birds and bats? For example, 9.9. Birds and bats have wings, but squirrels and lizards do not. So, are birds and bats more closely related to each other than squirrels to to squirrel or lizards. Before we jump to this conclusion, let us look at the wings of birds and bats more closely. When we do that, we find that wings of bats are skin folds stretched mainly between elonged fingers, but the wings of birds are feathery, feathery covering along the arm. The design of the two wings structure and components are thus very different. They look similar because they have common use for flying, but their organs are not common. This makes them analogous characteristics rather than homologous characteristics. It would now be interesting to think about whether bird arms and bat arms should be considered homologous or analogous fossils. Such studies of organs such can be done not only on current species but also on species that are no longer alive. How do we know that these extinct species ever existed? We know that from finding fossil fuels. What are fossil fuels? Usually, when organisms die, their bodies will be decomposed and be lost. But every once in a while, the body or at least some part may be in an environment that does not let it decompose completely. If a dead insect gets caught in hot mud, for example, it will not decompose quickly and the mud will eventually harden and retain the impression of the body parts of the insect. All such preserved traces of living organisms are called fossils. How do we know how old the fossils are? There are two components to estimation. One is relative. If we dig into the earth and start finding fossil, it is reasonable to suppose that the fossil we find closer to the surface are more recent than fossil we find in deeper layers. The second ways of dating fossils is by dictating the ratio of different isotopes of the same element in fossil material. It would be interesting to find out exactly how the method works. How do fossils form layer by layer? Let us start 100 million years ago. Some invertebrates on the seabed died and buried in sand. More sand accumulates and sandstones form under pressure. Millions of years later, dinosaurs living in the area die and their bodies too are buried in mud. This mud is also compressed into rock above the rock containing the earlier invertebrate fossils. Again, millions of years later, the body of horse-like creature dying in the area are fossil in rocks above these earlier rocks. Much later, by erosion of water flow wears away some of the rocks and exposes the horse-like fossil. As we dig deeper, we find the older and older fossils. Evolution by stages. A question that arises here is if complicated organs such as the eye are selected for advantage they provide, how can they be generated by single DNA change? Surely such complex organs will be created bit by bit over generations, but how can we each intermediate change be selected for? There are number of possible explanations. Even an intermediate stage, such as rudimentary eye, can be useful to some extent. These might be enough to give a fitness advantage. In fact, the eye like the ring seems to be a very popular adaptation. Insects have them, so does an octopus, and so do vertebrates. And the structure of eye in such of these organisms is different even from them to have separate evolutionary organs. Also, a change that is useful for one property to start with can become useful later for quite a different function. Features, for example, can start out as providing insulation in cold weather, but later they might become useful for flight. In fact, some dinosaurs had features, although they could not fly using the features. Birds seem to have later adopted the feature of flight. This, of course, means that birds are very closely related to reptiles since dinosaurs were reptiles. It is all very well to say that very dissimilar looking structures evolve from a common ancestral design. It is true that analysis of organ structure in fossils allow us to make elements of how far back evolutionary relationships go, but those are guesses about what happened in history. Are there any current examples of such a processes? The wild cabbage plant is a good example. Humans have over than 2000 years cultivated wild cabbage as food plant and generated different vegetables from it by selection. This is of course artificial selection rather than natural selection. So some farmers have wanted to select for very short distances between leaves and have bred the cabbage we eat. Some have wanted to select arrested flower development and have bred broccoli or the sterile flowers and have made the cauliflower. Some have selected for swollen parts and come up with kohlrabi. Some have simply looked for slightly larger leaves and come up with leafy vegetable called kale. Would we have thought that all these structures are descended from the same ancestor if we had not done it ourselves? Another way of tracing evolutionary relationship depends on the original idea that we started with. That idea was the changes in DNA during reproduction are the basic events in evolution. If that is in case, then comparing the DNA of different species should give us direct estimate of how much DNA have changed during the formation of these species. This method is now extensively used to define evolutionary relationships. Molecular progeny. We have been discussing how changes in DNA during cell division would lead to changes in proteins that we